Next lecture will be by Dr. Salim Elfishawi. From the National Cancer Institute, Cairo University. She's working. Where is he? Okay. She's working in the Nasser Cancer Institute and in bone marrow transplantation lab unit. And the talk is entitled Hematopoietic Stem Cell Transplantation, a Success Story from Genes to Therapy.
Hello everyone, thanks for inviting me. I want to thank the organizing committee for inviting me to say this talk. It's a very nice conference, and I will walk you through some of the issues. For all the past 3 days we were discussing problems with genetics and the hereditary disorders, and I think in this session of hereditary blood disorders, hematopoietic stem cell transplantation holds a curative therapy for all those with blood disorders that encounter the clinic. Hematopoietic stem cell transplantation aims at restoring the marrow function if it's immune or hematologic. It's used mainly in hematologic malignancies, acquired blood marrow failures, hereditary blood diseases, and it doesn't matter the source of stem cells. We can get them from the bone marrow. We can now move to the mobilized peripheral blood stem cells, and not so recently we adopted the umbilical cord blood stem cells. We can use either the allogeneic, the other donor, or the autologous stem cell transplantation from the same patient. According to the donor type in the hematopoietic stem cell transplantation, it's mainly divided into either matched, related, unrelated, haploidentical, cord blood, autologous, and syngeneic. I will go you through these types along with the presentation to know the difference between each one of them. When we say matched, we just mean the HLA region. Matching for the HLA region has been proven to be the most critical point in the success of hematopoietic stem cell transplantation. Those who know the HLA region, it's a very polymorphic region. It compromises many genes with the codominance inheritance. So the phenotype of the patient and the genotypes vary among populations, even within populations, within families. And so for the past 3 days you have all heard that we have a high consanguinity rate in Egypt. That's a bad thing that gets us all in all the genetic disorders and all the genetic malformations that we have. But maybe that's not a bad thing in Egypt and in Africa, because whenever you search for a donor, you want him to have a full match, 6 out of 6, that we imply for the 3 genes from the both maternal haplotype and the maternal haplotype for the A, B, and DR loci of the HLA region, and that you have a 6 out of 6 mismatch, and that doesn't happen elsewhere. You have 25% of having a 6 out of 6 mismatch, or a 6 out of 6 match in the HLA region. Difficulties we said about the stem cell transplantation would be the donor choice. We don't have that much donors, depending on the 6 out of 6 match in the HLA region. We have complications, we have morbidity and mortality, but the complications of stem cell transplants, mainly the infection, the graft versus host disease, and the veno-occlusive disease and mucositis. In Egypt, due to the high consanguinity rate mentioned by Dr. Temtami and everyone else, maybe this is the good side of it. We have a 60% chance of finding a matched sibling. And that makes us one of the luckiest places, because finding a matching sibling all over the world varies 25%, 30% maximum. So we did this study to find in 700 families, we found that we have a 60% chance of finding a matched sibling. And this leaves us with 40% of our patients lacking a matched sibling. When we looked at this 40%, we found that 36% of them had what we call a haplo-identical donor, that he is sharing 3 out of the 6 genes that they have. So, all over the world, the evolution of donor choice moved a bit from matched related donor, which is the family members, to what we call the matched unrelated donors. That's donor registries that unrelated persons would donate their bone marrow through registries, the cord blood bank, and the cord blood, and finally to the haplo-identical donor, where we have a mismatched family member that is 3 out of 6 matching at the HLA region. This has the advantages and disadvantages of each one of them. The optimum solution is the donor, the matched sibling donor, even they have 10 out of 10 match. With this optimum situation, we have a graft versus host disease in 25 out to 40%. When we move to the unrelated donor, we have the hurdles of the donor registries and the long time for finding a donor. The umbilical cord has so many issues in the finance of the blood bank, the cord blood bank, and we needing 2 units and the failure of graftation. The haplo-identical donor is one of the very nice recent adaptations for stem cell transplantation, because it holds the promise of easy access to the family member that's donating, the feasibility and the time management issues when you have to go for a retransplant or using a donor lymphocyte infusion for that patient. The syngeneic is the identical twin, but it's not that simple to find an identical twin to donate your blood to. When we went through the haplo-identical transplantation, we wanted to increase our odds for the success of this transplantation, because we have a very good chance of having the complications of stem cell transplantation, which are mainly the graft versus host disease and the infections. We try to move to a more robust mechanisms of finding immune cells that would help us in overcoming these problems. So we found those natural killer cells that are our assassins inside. They are very powerful cells that have powerful cytotoxic activity against viral infected cells and tumor cells. And we found that they were the first cells to recover post-transplant. So we thought of using these natural killer cells in this, the setting of haplo-identical transplantation to overcome the many complications of transplant-related morbidity and mortality. As you can see in this cartoon, the natural killer cells have what we call an alloreactivity against the T cells that caused us graft versus host disease and graft rejection, the dendritic cells that present the cells to the T cells, and they have leukemic, anti-leukemic effect against leukemia cells. Please mind, I'm always minded with leukemia because I work in the National Cancer Institute. We here also see a cartoon about the mechanism of the action of natural killer cells. Natural killer cells are adjusted to kill. They are in a continuous state of inhibition. When the cell is infected or having a tumor, they have a down expression of class 1, of HLA class 1 molecules, and then this triggers the natural killer cells to act. According to the natural killer cells, for them to act, they have this lock and key mechanism. They have their receptors and their ligands on the target cells. Their receptors are called key cells, among others. Those are inhibitory receptors. They were thought to be inhibitory, but now they were discovered that some of them are inhibitory and some are activating. According to their structure, they either have a short tail for activating receptors or a long tail for inhibitory ones. They have their ligands among the HLA class 1 molecules, the A and B and C1 and C2 epitopes. According either to they are inhibitory or activating, they start their actions. This was a revolutionary study that has been made in 2002 by Ruggery, that he was the first one to break the HLA barrier. He was the first one to transplant haploidentical siblings, and when he did so, he found that the natural killer cells, they have very high alloreactivity in this mismatched setting. And he concluded that the survival of especially AML patients has been 60% in the presence of natural cell alloreactivity. And this is another paper that has been recently published saying that you can even use this mismatching in the reduced intensity conditioning regimens. That's also for decreasing the morbidity and mortality of transplant-related complications from the conditioning regimens. We did our study here in Egypt for trying to elucidate the effect of KIR receptors and genotype on stem cell transplantation. Unfortunately, we didn't do it in the haploidentical setting because it's still new. We only transplanted 10 haploidentical cases, so we tried to use it in the matched sibling donor to see if this optimum situation also have alloreactivity. We used, I will go through the results. We had several outcomes, good outcomes with decreased relapse, decreased CMV infections, and improved platelet recovery and engraftments with the KIR receptors. Decreased survival was done to show that some ligands show decreased survival. One final thing about manipulating T cells and immune cells in hematopoietic stem cell transplantation. This is inducible caspase-9 gene suicide that is used to manipulate T cells that cause graft versus host disease. So we implant in them a suicide gene that we can turn on using a dimeric small molecules to preserve the T cell functions until they go to combat our viral infections. And when they cause us graft versus host disease, we finally push them to the apoptotic pathway. And these are this is the phase 1 and phase 2 clinical trials using the inducible caspase-9 suicide gene. I want to say in summary that evolution of genetics has enabled multiple donor sources coupled with manipulation in main immune response cells to combat against transplant-related complications. Immune therapy using natural killer cells and or modified T cells hold great promise in advancing transplantation. Thank you.
Thank you, Doctor, for your nice presentation. I think we have time for one question.
Thank you very much, Dr. Saleh, for the very interesting presentation based on your experience. I just wanted to ask two short questions. First of all, you said that you have a a very maybe restricted experience with umbilical cord transplantation, umbilical cord cells?
No. We don't have any experience in cord transplants because, first of all, for a successful cord transplant, you have to have 2 units. And here in Egypt, there is no
Yes, I
cord
understood
bank.
the obstacles, but I thought that at the beginning you said there's a limited experience. You mean a worldwide limited experience? I thought that.
No. The cord blood experience is now shrinking because it holds so much financial issues to construct a cord blood bank, and the allocation of the cord units to the patient takes a lot of time, and those patients. Not the hereditary blood disorders, they have the time in their hands, but for the leukemia patients, they don't have that much to find 2 matching units.
Okay.
And the graft failure rate is high with cord blood bank. So when they evolved to the family members again, and the mismatched actually haploidentical setting, this made it more feasible to eliminate the cord bloods or to decline their use.
Okay.
Because it costs so many, and we have the family members. Haploidentical family members include not only the siblings, the mother and father, and if he is an older patient, he has his children. So we have a lot of array of donors to search in.
Okay, but you